Alright guys, welcome to the very last video lecture for this year. I just want to start off by saying thank you guys for tuning in. I know again, there's lots of other things to be doing and there's, school's not always the first thing on your mind. So thank you for taking the time to better yourself and tuning in, watching the videos, and doing the work. Because that will help you out in the long run. And especially if you're wanting to go into the science field, I think this will really benefit you. And you may not see the benefits today, but you will in the long run. So thank you again for everything that you've done over the course of this semester and this crazy year. If you do have any questions regarding the phylogeny of the Beastie Boys, you can ask away in the chat, and I'll address those at the end if you'd like. I'll throw up the key if I can find it here later for you, and we'll kind of go over just a little bit of that. Today's lecture shouldn't be too long, just take a few minutes, but we're going to wrap up chapter 18. So last time we were talking about comparing patterns of development. Today we're going to kind of highlight some of the gene expression and how that's changed through embryonic development and also as species progress forward in their evolutionary history. So similar patterns of embryonic development develop as an outcome of highly conserved master genes. Again, these are the genes that are used as templates to code for developing embryos that make us into juveniles, and then so that we can progress into adulthood, become reproductive, and start the whole cycle again. But a mutation to the master gene can halt things or cause major problems, but it can also create new traits as well. A lot of times though, when you mess with the master genes, it's not a good thing. So you got homeotic genes encode for transcription factors, and you've got the example there, the floral identity of apetal 1. Like I mentioned that last time, that would cause where normal petals on the flower would form. Instead, you'd have like the stamens coming out of there, which are the reproductive parts of a flower that produce pollen. So instead of petals, you have stamens that are going to produce more pollen. It's kind of weird. So messing up these master genes really affects the physical appearance of the organism overall. Developmental comparisons in animals. The embryo of many vertebrate species develop in similar ways. If you looked at the textbook, you might have seen that there are certain stages that a fish, an amphibian, a chicken, and a human all share in common, which is kind of neat. So there are certain Hox genes that determine the genetic body plan. So as you develop, certain things are made at the right time. Differences are brought about by variations in gene patterns. We talked about some of those examples. So here's this awesome picture of the different embryonic stages developing. As you see, the salamander, the human, the rabbit, the chicken, and the fish. As you look at the very first stages at the bottom, we all look very, very similar according to the pictures. As you look at the actual things up there, there's a lot of similarities. A lot of these all have tails. They have some type of head region. They have these gill slits. It would be very difficult to distinguish them just by looking at them because of so many unique traits that they have in common. But as they progress further into the embryonic development, we start to see certain things emerge, maybe limbs, or the tail remains for, like, say, a salamander, but not a human. So different things or genes are turned on and turned off, and they have changed over time. They've either duplicated, had other mutations, whatever it may be, and that distinguishes us differently from other species as well. But the Hox genes are homeotic genes of animals. The pattern of the expression of Hox genes determines the identity of particular zones along the body's axis. So as you can see, each little axis within this particular pupa stage will turn into the fly's body plan, and that again is encoded for by the genes at the top, the Hox genes. So if you take a gene and you move it or you delete it out, this whole body plan may be non-existent. And if that's non-existent, that means the organism probably will never be there. Okay, so there was a question by T here. Are the gills we see on the humans fetus our ribs? It turns out no. The gill slits that we see, and they're not actual gills. Keep that in mind. We never have the ability to breathe oxygen through the water. They're just slits. Like on a fish, you have that opening where the gill is, but as you look inside a fish, those red little things, those are the gills. So we just have gill slits, just like, if I go back this page, salamander, human, they have them, but over time, apoptosis will delete those things out. Those cells will actually be destroyed, and new tissue will form. The ribs, believe it or not, come later, and the ribs are actually controlled by a Hox gene as well. And the book, it actually tells you that the Hox gene is coded for is by HOXC6. I think that mentions it here in a second. But the HOXC6 gene actually determines along the vertebrae where the ribs will actually form. So if you notice, like on your neck, you don't have ribs. And if you had a tail, like a dog, there's no ribs forming on the tail. The question is why? Is certain Hox genes are located in those areas, in those body plans, as you can see, like in the pupa stage, that will start that growth of those bones, which is kind of cool. So a really good question there, T, about the ribs and the gills. They did some experiments with fruit flies, as I mentioned last video, with the antennae. So they actually duplicated it, put one of the legs in the antenna's part, and the fly actually grew legs out of its face where the antenna should have been, which is pretty crazy when you think about it. If you had ears, for example, and we duplicated the gene for your arms, the budding of your arms, you could have actually arms on your head. That'd be absolutely ridiculous. Not saying they'd be fully functional or anything like that, because you have to have a shoulder socket and stuff like that. But how crazy would that be, that you had legs coming out, or arms coming out of your head? So we know that this can happen, and we can do things like this. Now, again, we go back to the ethical standpoint. Would that be ethical to put, legs or arms on somebody's head? No, not a human being. Fruit flies, they live about 3 or 4 days, and nobody cares about insects. So there you go. So vertebrate Hox genes. Invertebrates expression of the HOXC6 gene for the ribs, once again, develop on the back and not the neck or the tail. So that again determines your body cavity, and it doesn't code for ribs higher up or lower in your body. It'd be really weird if your body was just like one giant torso, just all the way up to your chin. When you think about it, that'd be like a giant worm, I guess. So I'm glad we're not worms. The DLX gene encodes for a transcription factor that signals the embryonic cells to form buds that give rise to your appendages, which means your arms and your legs. So we have the arms that are great for us to move around and do stuff, but they start off as little bitty nubs, just a cluster of embryonic stem cells that start growing, which will later develop into your forearms and your humerus and your fingers, all that stuff. And when we went in for some ultrasounds when we found out we were having a baby, the first couple pictures that they take is a little bitty thing, and there's not much to see. And it was really funny because I was like, it doesn't even look like a kid, it looks like a gummy bear. When you think about a gummy bear, they've got the little nubby arms. That's exactly what the child looked like inside there in the womb, just like a little gummy bear. It was really funny because she started wiggling and dancing, so she was a dancing gummy bear, which we got a kick out of. So Hox genes suppress the DLX, once again, expression in all parts of the embryo except for where the appendages need to be. So this is why you don't typically have a third arm growing out from your hip or something like that. It's because those Hox genes are going to be suppressed so that you don't have extra things. And you're thinking, well, would it be necessarily a bad thing to have an extra set of arms? Maybe not, but again, you have to have a shoulder joint to actually move them. So if you didn't have that anywhere else on the body, it would just kind of gonna dangle there. So it wouldn't be really productive. So again, as you look at this, you can see a human being right there, but on the other side you have the snake embryo, which is kind of cool. And now the snakes, when you think about a snake, they're very long, and they actually have numerous ribs. So as you highlight in the human where development for ribs is going to be, it's just right here along the back region. Okay, and even then, when you think about your ribs, it doesn't go all the way down to your hips. They actually stop before then. But as a snake, they actually have ribs that go all the way down, and it's highlighted in purple there. So they have numerous ribs within a snake, and that's a lot of times to help them breathe and constrict their body and stuff like that. So they have a gene that's not suppressed in their body. Okay, so persistent juvenile features. Sometimes organisms evolve, and as they change, sometimes they just look like a younger version of the other species, which is kind of cool. So some of these juvenile traits tend to persist in certain newer species for whatever reason. And it's not always the case, but some do. So a chimpanzee skull and a human skull, they actually look a lot alike when you just hold them next to each other. So a baby and a baby chimpanzee just look about the same. As they develop, though, the skulls change quite a bit, and you can actually hold a chimpanzee's adult skull and a human skull, and you go, well, yeah, I can tell this is chimpanzees. The chin shape changes, the broadness of the forehead changes, lots of things just change between the skulls. However, they believe that many of the primates that were long ago, our ancestors, some of them might have had some more of these adult features, but the human population tended to persist with these younger features for whatever reason. And that's why we today look quite a bit different than our other relatives that are other primates. We don't have as broad a jaw, and we don't have some of the other features that are found in the skull. So maybe a little more size for brain power or whatever it was, but that's what happened. Okay, so in this picture here in the book actually shows you that. As you can see, the infant of the chimpanzee looks a lot like a baby's, as in humans, but you got that much broader jaw, the skull just a little wider here through the face. But as the humans, you can see babies look about the same. And again, humans, we just haven't had that broad jaw and whatnot. And evolutionary speaking, they talk about how our jaws as humans are getting smaller. And you're thinking, really? How they know that? The wisdom teeth. Wisdom teeth used to come in, they think, back in the day just fine. That our jaw was much larger, accommodated for that space. But as we've evolved over time, our jaws are getting much smaller. But the wisdom teeth are still coming in. And they typically come in at angles sometimes for people with smaller jaws, which cause pressure pushing on the teeth in front. And it's very painful. And I know some of you in class were talking about your wisdom teeth. I too had my wisdom teeth pulled because they were causing problems. And as such, that's a common thing for a lot of people to have them pulled because there's not enough room for them. So there you go. Persistence of juvenile traits in salamanders. The axolots, these external gills and other larval traits persist in adulthood. So these little guys are kind of like salamanders, but they mainly stay in water because they have these external gills, these little fuzzy things. It looks like hair. Those are actual gills, but they're external gills on the outside. They're kind of like a fish, but theirs were not deleted by apoptosis for whatever reason. Other salamanders, they can venture onto land and breathe air, which is perfectly fine, but these guys retain that juvenile feature, which is kind of cool. But there are, like I said, just different aspects of different organisms based on their environment, what they need most. Take-home message: why our similarities in development indicate a shared ancestry? Again, similarities in patterns of development are the result of master genes. And master genes are typically conserved through evolutionary time. Some differences between closely related species are a result of a master gene mutation, duplication, deletion, whatever it is, and it usually changes the development patterns. And that's why we get some of these differences in physical features. But what we do is we can use phylogeny to look at these genes and how they've changed over time to determine how closely related they are to one another. And we can make some predictions. We can do all sorts of cool things. All right? We use this information about phylogeny to understand how to preserve the species that exist today. And you use the GenBank website, which is in conjunction with the NCBI website. So you can look at nucleotide sequence, protein sequences, whatever it is. You guys built your cladogram phylogeny for species, a particular gene, whatever it was that you chose, using that website. All that's free to use, and it's all out there, public knowledge. You can see it. So the more DNA we put out there, the more we can see how genes change. We can see how species have changed over time. And they apply that to, say, conservation biology with those honey creepers on Hawaiian Islands. Again, many of them started going extinct due to organisms invading their natural habitats, competing for the same resources. But what we do know is that these birds, as we start looking and sampling the DNA from them, we can determine how closely related these populations are and determine if they're actually inbreeding. So maybe there's a population on one island of Hawaii and the other population on another island. Maybe we could take two of those, put them together, and they can diversify their genetic diversity. Because otherwise, if they're all inbred, it leads to what is called inbreeding depression, which usually means there's not as many genes out there, which means they're not very adaptable. A virus, a disease could wipe them all out, kind of like bacteria. They're all the same, they all get hit equally. But if we can figure out this, we can actually write down their code. We can take cells, freeze them. Perhaps one day we could actually clone these birds and bring them back, which would be kind of cool. So the more data we collect, the more DNA we collect, it is a possibility we could synthesize these DNA strands and or clone them in the future. Which is pretty crazy. And that's what they're working with, with the woolly mammoths. They'd like to find enough DNA to bring back one of those. Again, we talked about that in class 2. So there you go. Those are the honey creepers. Again, no, they did not get these birds to position like that just because of a photo shoot. They are dead stuffed birds, so they're collections of birds to show the diversity. They do have cells and DNA that they could possibly use. There's usually a culturing process for the taxidermy on these. They inhibit that DNA from staying intact. But this is the evolutionary history of how these honey creepers were related and how they are currently related. So they're showing the genetic diversity between all of them. All right, conservation biology continued. Cladistics and analysis are also used to correlate past evolutionary divergences with behavior and dispersal patterns of existing populations. We can look at mitochondrial DNA sequences that suggest the blue wildebeest populations are genetically less similar than they should be, which is kind of cool. So we didn't know that, but we did after testing their DNA. Using a combination of data, conservation biologists can recommend measures to improve gene flow. They also did this with cheetahs. So cheetahs, of course, got hunted for their furs, because they're very beautiful furs. Dropped them down, got down to a very few, select few left. So they realized that they're breeding with their brothers and sisters, inbreeding depression, incest, all that, not good. So they're trying to find different populations of cheetahs and then hopefully move them to new areas so that they can mate and increase their genetic diversity over time. So lots going on for conservation biology, keeping species alive, usually in part to human impacts. Medical applications. Researchers study the evolutionary infectious agents by grouping their biochemical characteristics into clades. The example that they gave in the book was a phylogenetic analysis of the H5N1 influenza virus isolated from pigs. Showed that the virus actually jumped from birds to pigs. So there, if you recall back in the day, there was the H1N1 flu virus. That was something that hit right around 2008, 7 or 8, somewhere in there. But again, this is very similar. It's a variation of it. So there's a bird flu that jumped to pigs, and it jumped from pigs to humans. So yay! But it did this a couple times. So when we think about the COVID-19, my first reports it came from bats, and then it went from bats to people. So yes, it can jump from mammals to mammals depending on how closely related are and how infectious these agents are. So again, you have COVID-19, but there might be another COVID out there variation that they've been studying as well. But as viruses and bacteria move along, they can evolve and change just like species do, but usually at a much quicker rate because they're much smaller and they can diversify off. So, yes. All right. Take-home message from that: phylogeny research is yielding an ever more specific and accurate picture of how all life is related by shared ancestry. Among other applications, phylogeny research can help us preserve species in danger of becoming extinct and to understand the spread of infectious diseases as we see today. All right, guys, that is all I had. If you want to take a look and stick around for the key on the Beastie Boys, I'll pull that up for you here real quick. So you might have had a few different traits than I did that I asked you to fill in the last couple. So if you named them differently, that's no big deal. So don't feel like you have to change it to what I have on here. So if what you have is a different wording, that's okay. All right. So these are the little beasties that you had to identify and pick out the traits. These are the ones that I came up with, and there's only 9 of them. So you have the eyes, eye size, teeth, antenna, a fin as I call it, feet, tail, spots, a beard, and tongue. You might have called it something else than a beard or a tongue, whatever it is. Okay? But those were what I had. So again, the outgroup needs to have none of these things. So we wanted the outgroup to have zeros all the way down, and then all the other organisms to have evolved these traits over time. So this is your taxon by character matrix. And I wrote it out this way because the outgroup, once again, had the least number, zeros across the board. And then we get to E, and it only had 2 of them. It had the eye size and the tongue. So something that was very similar to E. All right. B, as you look at it, had a few more. But again, they had 4 traits. This one also, F, had 4 traits. But again, they weren't the same 4 traits. So yes, they can have the same number, but they should have different traits total to distinguish them as different species. If they were exactly the same, if B and F had ones here and here, then that means they're the exact same. And sometimes that does happen when we look at genetics and or species or characteristics. We go, well, turns out they're the exact same. Or you just didn't have enough characteristics to distinguish them differently. Maybe ones, they look exactly the same, but one has a small black dot on it. Maybe you didn't account for that character trait. But again, as you look at it, you should have something similar to this. The outgroup way down here, and then you have something that looks like this. Now, again, if yours didn't look like this, that's okay. All these branches can actually twist, and we would have gone over that a little more in class. But they can twist, turn however you need to. So if you have CNA swish, that's okay too. But yes, you have the tongue evolving in certain areas. You have the big eyes, spots from D and B, the beard only in B. You've got branched antenna, you got a bushy tail, pointed feet, all of which. Okay? So I'll leave this up here for a little bit and see if you have any questions regarding the Beastie Boys assignment. But otherwise, you are done. Okay? But again, thank you guys for being awesome and doing all the things that you've done these last couple months for me. I really do appreciate it. That says a lot about you and your character as a person. Alright, I don't see any other questions, so I'm going to cut you guys loose. Take care and enjoy that summer. Thank you again.